Okay, welcome to the LOC Symposium, The Constructive Brain. Ever since the pioneering work of Hubel and Wiesel, we have studied the brain in a conceptual framework in which features can be detected as a function of changing features in the outside world. The brain is responding to it. The receptive fields on the visual systems are specific to the features in the outside world. And that also gave a conceptual framework to study the brain, which sometimes over the years was a bit simplified or simplistic, even though never intended. It gives rise to the idea that these neurons and the brain in general is kind of in a waiting mode, silent, waiting to be switched on. And the stimuli that are presented are the kind of awakening signals that click the brain on. And of course, that's a simplification. We know that's wrong. If anything, it's just a ripple on top of ongoing activity. So there's a lot of activity in the brain, lots of energy consumed during the state in which specific neurons, for example, aren't switched on. And so there's a conceptual question, what is happening? And so within this LOC Symposium, we are looking into a more inside-out perspective to ask the contribution or the brain activity before it is stimulated. And in principle, there are kind of three ideas. One, it is contextualizing perception. And the other one is memories started in the brain, so it's the inside-out perspective. And the third one is that it is contributing to prediction of the outside world. And so these will be, in general, the three themes that we cover. We start with a multi-scale approach going all the way from rodent neuroscience to non-human primate. And we start with Matthew Self, and the title is Isolating the Information Conveyed by Feedback Connections in Monkey, Mouse, and Mouse Visual Cortex. Welcome, Matthew.
Good morning, everyone. So first of all, I'd like to thank the organizing committee for arranging this nice symposium and inviting me to come speak here. I'm going to be talking about isolating the information conveyed by feedback connections in monkey and mouse visual cortex. So I'm aware it's organization for human brain mapping, but hopefully during the course of this talk, I'll make it apparent how this relates to human brain imaging. So my particular interest is in the function of feedback connections in the brain. So we know that information travels in two main directions in the visual system. So here we see on the slide a macaque-monkey brain. And feed-forward connections in the visual system travel from the retina through the thalamus into the primary visual cortex at the back of the brain. And then from there forwards into the higher visual areas, such as areas like V4 and IT. And these feed-forward connections arrive in layer 4, and they are strong driving connections which cause their target neurons to fire. And not only that, they define the functional properties of these target neurons. So they tell a neuron where it should respond to in space and what sort of features it should respond to, be that a oriented line in V1 or perhaps a particular face in IT. But there are just as many connections going in the opposite direction, so from the higher visual areas back down to lower visual areas. And it is these feedback connections which have a much more mysterious function, so we don't know exactly what they're doing. But I think most people would agree that feedback connections, well, at least part of their function, is to convey information about the brain's internal perceptual model of the world. But exactly which information is conveyed and how feedback affects ongoing activity in early visual areas is still a matter of intense debate in the field. Now, one of the reasons it's so hard to figure out what feedback is doing is because it arrives very early in early visual areas, and it does interact very strongly with feed-forward activity. And so a trick that researchers have used for many years is to actually isolate feedback from the feed-forward effects by using occlusion. So on this slide, we have a very famous image, the front cover of the Abbey Road album by the Beatles. And you might be able to see a subtle occluder that I have added to the image. So this large gray rectangle. And you could probably know who I've occluded and why I occluded him. So the trick here really is that by occluding part of the image, you have removed the feed-forward input from that part of the visual scene. There is no change in retinal activity in the occluded region of the scene. However, therefore, if we see any activity changes in that region, that these must be coming from other sources, from horizontal connections or from perhaps feedback connections. Okay, now, by then looking at what happens in this occluded region, we can therefore focus in on the function of these feedback connections and what sort of information they are bringing back to the early visual areas. And we can ask two interesting related questions. So first of all, are the feedback connections bringing back information about what would be predicted to be there? What is behind the occluder? Or in other words, do we get any Paul McCartney information in this particular image coming into the occluded region of the scene? The second related question is there any information in the occluded region of the image about the rest of the image? So would you get any information about George or Ringo in the occluded part of the image? And by answering these questions, we can help to try and understand what feedback is bringing back to the early visual areas and what possible function it might have. So I'm going to start with this second question. What information is there in the occluded region about the rest of the visual scene? Now, Lars Muckli, who introduced the session, has for many years used this occlusion paradigm in humans using fMRI. So in this study, they're typically using a natural scene, one of maybe 3 natural scenes, and the bottom right corner of the scene is occluded. And it's been shown many, many times that you can easily decode the information present in the rest of the image from the voxels that are in the occluded region of the scene. And this particular result comes from the 2015 paper where Lars did this in a laminar resolved fashion. So here the important result is the green line, where they showed that decoders which are built in different layers of cortex are able to decode the activity in the rest of the scene. And in particular, decoders in the superficial layers of cortex were the best at decoding which image was presented to the subjects. And these are the layers which receive mostly feedback connections in the visual system. Okay, so it does therefore seem in bold MRI that you can decode what is in the rest of the scene from the occluded region. But of course, there's the caveat here. This is bold. So bold doesn't necessarily correlate with the underlying spiking activity. And furthermore, we know that the bold signal is very sensitive to top-down connections. So if I was to actually put an electrode into V1 and look at the spiking activity there, would I still see responses to these occluded regions? And would we be able to decode the rest of the image from the occluded spiking activity? So that's exactly what we did. So we used an occlusion paradigm in monkeys. We did exactly the same sort of paradigm that we used in the humans with Lars's work. So in fact, we used exactly the same images that they used in this study in 2019 by Tyler Morgan. So Tyler kindly provided these images to us. These were 24 natural scenes, things like beaches, forests, but also things like industrial scenes and buildings. Now we showed these either non-occluded or with the bottom right corner occluded. And this was a very easy task for the monkeys. They just had to fixate on a fixation point, and then we showed the stimulus for 700 milliseconds. We recorded multi-unit activity with chronically implanted UTAR arrays in V1, and this was recording multi-unit spiking activity. And we were very careful to select only receptive fields which were in the bottom right quadrant of the image, and only those which were at least 2 degrees away from the boundary between the occluded and non-occluded region. So this is the response you get to these occluded and non-occluded images. Now this is not decoding or anything, this is just the basic spiking response of the cells. And what you can see first of all in blue are the response to the non-occluded images. And each line here is a response to one of these 24 images across cells. And what you can see is very, very rapidly, the responses diverge across these 24 images. So we can actually measure the time at which the responses to different images become different to one another, and that happens at around 46 milliseconds. Right, that's not too surprising here. There are different visual features in the receptive field for these different images, so of course the responses will be different. What's more interesting is for the occluded images. There, first of all, what we see is actually a rather weak response, right? So certainly the response is nowhere near the height you get for the non-occluded images. And there's this characteristic excitation, inhibition, excitation temporal profile. But there are differences between the responses to the different images. And again, we can measure the time at which these responses start to diverge across images, and that is around 85 milliseconds after stimulus onset. So there is this delay before the occluded information starts to come into cortex and before it tells you something about what's in the images. Now, of course, the fact that these lines are different doesn't mean there is actually information about what's in the rest of the scene. So to determine if there is information, we used a decoding approach. So here we trained linear discriminant analysis decoders. These are multi-class. They tell you which one of the 24 images was being presented. And what you see now on the y-axis is the decoding accuracy, and this is done in a temporally resolved way, so over time. Now, for the non-occluded images, the decoders go from chance performance, which is about 4%, all the way up to ceiling performance almost immediately. So they go straight up after about 40 milliseconds. Again, it's not too surprising. There is visual information in the receptive field. We can decode it very easily. For the occluded images, it, again, is slightly delayed. It takes some time, and the decoders only reach ceiling performance at around 100 milliseconds after stimulus onset. But we are able to decode the rest of the scene from the spiking activity in V1. So we already answered our first research question. Yes, the spiking does contain information about the rest of the scene in the occluded region. Now, we can dig into that a little bit more deeply, trying to find out what sort of information is being sent back to V1 here by looking at cross decoders. So in pink, we see a decoder that was trained on the occluded data and then tested on the non-occluded data. And you can see that one does reasonably well. It's well above chance. Whereas the non-occluded trained and then tested on the occluded does rather poorly. Now, the reason for this, we think, is that when you train on non-occluded data, there are visual features in the receptive field. You know, there are orientations, there are contrast elements. And that's what the decoder picks up on. These are strongly driving the cell. So when you show an occluded image, those elements aren't there. And apparently what is being fed back to V1 is not the very local individual elements that should be in the receptive field. Right? So the decoder fails. Whereas in the pink case, when you train on the occluded data, there are no visual features in the receptive field. So the decoder has to pick up on something else. It picks up on what is being fed back to that region. Now, we don't know exactly what that is, okay, but people have suggested it might be something more like a rough sketch of the image or something related to the gist of the image. But it's something that is also there in the non-occluded state, and so we are able to do some cross-decoding. Now, we can also, just going to skip over that, we can also look at this decoding in a slightly different way by comparing it to what happens in humans. So in human data, we actually can compare the images by putting them into the same framework. So if we look at Tyler's study, he showed these exact same 24 images, and we can compute representational dissimilarity matrices. Right, so I'm sure many of you are familiar with these, but each point in this matrix reflects the dissimilarity between the response to a pair of images across all the voxels in V1. Okay, so dark points would be a pair of images that are very dissimilar in their response across voxels, whereas light squares would be a pair of images which produce a very similar response across voxels. Okay, and that gives you kind of fingerprint about how V1 is representing this occluded information. Now, we can do exactly the same in monkeys, but we can do this in a temporally resolved way, right? We can do this for every single time point in the data because we have very high temporal resolution. And now what we can do is actually compare the monkey and the human data once we've put it into this same framework, right? So we can do a pixel-wise by pixel-wise Spearman's rank correlation between these matrices and see, is the informational structure that's being sent back to monkey and human V1 that's having the same structure? The result you see on the bottom, and that shows that initially there are no correlations between the monkey data and the human data. That's not surprising, right? Before the stimulus comes on the screen, all the monkey responses are very, very, very similar. But after about 100 milliseconds, you see these correlations reach very high levels. In fact, they're close to the ceiling sort of levels that you might be able to achieve given the noise in the data. So this shows something which I think is kind of remarkable, that what we're seeing here is that despite the difference in species, despite the difference in technique here, the information that is being fed back in the spiking in V1 has a very similar information structure to the information that's being fed back and is detectable in the bold signal in humans in V1. Okay, so now I'm going to switch over to a different species to carry on look into what is being fed back to V1 in the absence of feed-forward information. So we're going to turn to mice. In mice, we had head-fixed mice, and we were first doing electrophysiological recordings in V1, but we're using slightly less naturalistic stimuli than the ones we were using before. So these very simple grating stimuli, where we either had stimuli in the receptive field or a uniform stimulus across the whole screen, or an occluded image again. So we have this gray circle which was centered on the receptive field. Now, we were very careful to ensure that the actual occluder was centered on the receptive field and that the 100% of the receptive field was inside the occluded region, right? We don't want any of the background information to get into the receptive field in a feed-forward way. And we could see very clearly that there was no feed-forward activation of the receptive fields in the occluded state. So what you can see here, for instance, in the black and the gray lines are the visual responses caused by the classical stimulation of the receptive field. Whereas in the red line is when we have the occluder. And what you can see is, again, it takes some time before the occluder actually activates the cells. And this first initial burst of spiking that you see in the other conditions simply isn't there. And that was true across the population of cells that we recorded from. Now, we can also see that by looking across the different layers of V1. These were laminar electrodes, and we could look across the superficial layer 4 and deep layers of cortex, that's what you see on the y-axis, across time. And normally, if you show a visual stimulus in the receptive field, this will activate all layers, and you get sustained activity in the superficial and layer 5. But if you look at the occluded information, you can see it's like somebody cut a hole in layer 4, and that there was actually no initial spiking activity at all in that layer. I think you can see that even more clearly if you look at current source density, which shows you the current flow that is going on in cortex. Normally, when you show a visual stimulus, you get this big current sink as the input comes into layer 4 and drives current into neurons. So that's this big red blob that I've arrowed on the slides for the classical stimulation and the uniform stimulation. But when the blanks situation, when we have an occluder, that initial current sink is completely absent. Okay, so here the only point is really that there is no feed-forward activation of the cortical microcolumn using these occluders. And other groups, for instance, the Scanziano group, have shown that if you inhibit the high visual areas in mouse visual cortex, then you can also completely remove these contextual effects. Now, next we were interested in a particular puzzle, which is why is it able to drive spiking activity in visual cortex? We normally think of feedback as being rather a modulatory signal, a weak signal that is coming into the apical dendrites of cells in V1, and only really able to drive cells if it is coincident with feed-forward activity. So if you remove the feed-forward activity, how can you actually drive the activity in using feedback? To look at this, we suspected that what might be happening is that in occluded states, that the cortical microcircuit might be disinhibited. So we started looking at different interneuron subtypes in V1, because there is this well-known disinhibitory circuit within the different interneurons of primary visual cortex. So it's well known that pyramidal cells are the excitatory cells in cortex, and they receive inhibition from parvalbumin interneurons, PV cells, and from somatostatin interneurons, more to the apical dendrites. But interestingly, there is a disinhibitory motif where VIP interneurons actually inhibit somatostatin interneurons. So that prevents the somatostatin interneurons from inhibiting the apical dendrites, and that releases the pyramidal cell from inhibition. So we hypothesized that we might see more activity in VIP interneurons in occluded regions of the scene, which would perhaps allow the cells to become more responsive to feedback input. So here we see the responses to different cell types. So on the left, we have the pyramidal cells, the excitatory cells, then parvalbumin, VIP, and somatostatin. Now, what we saw is that, first of all, if we look at the excitatory cells, they respond very well to uniform stimulation, but also very strongly to these contextual stimuli. Again, it was slightly delayed, but there's still a strong response here. Much stronger than in the monkeys, perhaps because we used a smaller occluder here. The parvalbumin neurons do pretty much exactly the same thing as the excitatory neurons. But VIP and SST, or somatostatin, do something really quite different. So for VIP neurons, the contextual stimulus is the favorite stimulus of a VIP interneuron. It responds extremely strongly to a contextual stimulus, to a stimulus where there is an occluder. Even more so than it does when there is actually information in its receptive field. In contrast, the occluded image is the least favorite stimulus of a somatostatin neuron. It's actually suppressed when you show an occluded image. So we think this shows strong evidence for a disinhibitory circuit here, where when you show an occluded image, VIP interneurons are strongly enhanced. They suppress somatostatin cells, and that releases the pyramidal cells from inhibition. And that allows them, therefore, to be more sensitive to feedback inputs coming into their apical dendrites, and might allow feedback to actually drive spiking activity. We tested this causally by actually optogenetically inhibiting VIP interneurons. So we put an inhibitory opsin in the VIP cells, GT-ACR2. When we shine light on cortex, this silenced the VIP interneurons. And what we found is when we did that we were able to reduce the level of this contextual effect, this occluded effect, to about 60% of its previous level. Okay, so it seems therefore that normally VIP interneurons are amplifying up the response to feedback connections. But we couldn't get rid of all of the effects, so perhaps this might be an experimental thing that we couldn't silence all of the VIP activity, or perhaps there are other mechanisms by which feedback can still drive activity even in a non-disinhibited cortical microcircuit. Now, the final thing we wanted to look at was to look at the information that was present in this feedback signal in the mice. So in each of our experiments, we used 6 orientations in the background. And we had many cells which are orientation tuned. So here's an example of a classically orientation tuned cell. This cell likes something around 80-degree orientations. And to analyze this data, we took the orientation tuning of each cell and we shifted it to 0 degrees. So 0 degrees became the preferred orientation of the cell. And then we recoded the data so that into the difference from the preferred orientation. Now, what you can see if you do this is that for uniform stimuli, of course, you can get a very, very strong response to the preferred orientation of the cell, which very quickly decays away as you move the orientation away from the preferred orientation. And it comes down to close to spontaneous levels. But for the blank stimulus, for the secluded stimulus, actually it's quite different. It responds reasonably well to all of the different orientations in the background. Right? So it almost doesn't matter which orientation is the background. These cells will all respond. There's only a very subtle orientation tuning left in this curve. So you can probably see the response at 0 is slightly higher than the response to the other orientations. And this was significant, but still rather subtle. So this seems to argue against the idea that feedback is really bringing back the specific feature that should be present in the receptive field here, where we'd expect to see most of the activation at 0 degrees. Instead, it seems to be more of a subtle bias across the population. So that raises the question, could we even decode which orientation was in the background in this particular case? And the answer to that is yes, we can. So we can run these same classifiers again that we used in the monkeys, and they work perfectly well. Okay, so it doesn't matter whether you train on uniform, test on uniform, train on blank, test on blank, every combination gives you above chance decoding. So it seems that although there are only subtle biases in activity, they are enough to drive these decoders and allow you to decode what is present in the background. And these decoders are definitely picking up on orientation-tuned information. If you look at these confusion matrices, where we look at what the classifier chose based on the actual orientation that we tested at, you can see that when it does make a mistake, it tends to make a mistake at a neighboring orientation. Now, interestingly, we could also look at which cells were most informative for these decoders, so which cells carried the most weight in the decoder's decision. And actually there we did find cells, a small population, something like 4 to 5% of cells, which did show very strong orientation selectivity, even when there was nothing in their receptive field. Right, so here you see the response of a cell in gray when it is being stimulated by visual information, and in red when the receptive field was blank. And you can see it gives a very strong selective response to the background orientation. So what we suspect might be happening then is that on top of this weak bias that is present in many cells, there is a small subpopulation of cells which does contain selectivity about what should be in the receptive field if it wasn't for the occluder. So to kind of wrap that up into one slide, here we have an occluded region of V1. We have cells which prefer different orientations, and these have different colors in this image. And when you show an occluded image, you will activate all of these cells, right? It doesn't matter what the orientation is in the background. But there's a subtle bias, and the blue cells, probably almost too subtle to see here, the blue cells are going to increase their activity slightly as they prefer the orientation that is in the background. On top of that, we have a small subpopulation of cells, these supercells. We don't know what they are yet, but we're very interested in trying to find out, which do actually strongly respond to the background orientation. Okay, finally, one last thought about what might be happening to the occluder. Actually, I'm going to skip that, I think, in the interest of time. So I'm just going to give you my takeaway messages. So when the images are included in the brain must represent two things at the same time, the occluder and the included information. Information about these two things are fed back to V1 and affect spiking activity in this region. The majority of cells in V1 actually represent the actual occluder itself, the gray circle. And the strength of this representation is dependent on the perceptual organization of the scene. But the occluded information produces subtle biases in the large majority of cells and larger effects on a smaller subpopulation of cells. So finally, just the acknowledgements. I'd like to thank everyone at the Netherlands Institute for Neuroscience who took part in this. So Paolo, Feng, and Singh, who did the monkey work, Lisa, Sridip, and Peter, who did the mouse work, and the team at the University of Glasgow, Tyler, Lucy, and Lars, for providing us with the human data and for all their discussions about what this might mean. And finally, thank you for listening.
Thanks a lot, Matthew. There are a lot of good questions, and now, yes. And I'm afraid we won't go through all of them, but I will start with a highly ranked one. Is the information in the occluded region really due to feedback or crosstalk between neighboring neurons, possibly from the non-occluded region?
Yeah, my slides just disappeared, otherwise I have an extra slide to show you that. So we looked at that by actually, here it goes. Oh, okay, no, apparently not. Okay, so what we did is we isolated and we built decoders based on cells which were further and further away from the occluded region. So we built new decoders by only using subpopulations of cells, depending on their distance to the occluded region. And what we found there is that essentially you could decode at all the different distances from the occluder, and the latency at which activity started to change did not depend on the distance of the cell to the occluded region. So what we think is more likely is that this is really a feedback effect, and that feedback is coming back en masse to the entire occluded region, and not spreading from the edges of the occluder.
And a quick question, how well do you get monkeys to fixate?
Really well. So our monkeys are fixating in a 1-degree window, and we actually even have a smaller window within that where they have to trigger the trial. And they have to stay within that 1-degree fixation window for the entire period of the stimulus. So there's no possibility that the eye movement could bring the occluded information into the receptor field of the cell. And for mice, it's slightly different. For mice very rarely actually move their eyes when they're head fixed. So for them, we throw out trials where they do move their eyes, but otherwise they keep their eyes in this sort of constant position throughout the trial.
Does it make a difference whether or not the images are behaviorally relevant for the monkeys and mice?
Yeah, that's a really good question. That's something we're actively researching together with Christian Levert's group at the Netherlands Institute for Neuroscience. So they're training mice to detect these images or subsets of these images. And they find really dramatic changes in activity in pre and post training data. So it does seem to make a huge difference whether the stimuli are behaviorally relevant or not, but I can't tell you very much more about it at this stage.
Does the occlusion paradigm provide clues about feedback looking like a backpropagating algorithm?
Can you say that again?
Does the occlusion paradigm provide clues about feedback looking like backpropagating algorithms? So,
yeah,
it's more a computational question.
Yeah.
Computation network. Actually, we in our lab have something which is not a backprop network, but deep convolutional network.
Yeah, so I'm not sure I can really speak to that question particularly well. So, I mean, what we see is that we don't see much evidence that feedback is coming in this particular case and subtracting away information in the superficial layers of cortex. So we see more of it seems to be an additive term in the visual cortex. So it's adding data to the superficial layers in particular. I'm not sure if that gets through. Yeah.
Thanks a lot.
Thanks.
So next is Peter. Yes. Next speaker is Peter Koch, the neural circuits underlying subjective perception. Thank you.
Great. Thanks everyone for coming, and thanks for the organizers for inviting me, especially Lars. It's a great symposium to be a part of. So I investigate the neural circuit underlying the way we perceive the world in humans, so using human neuroimaging, so not as precise as the data that you've just seen, but hopefully still something of interest. Let's see if this slide clicker works. Yeah. Okay, so this is let's start with an example. This is a statue on the carving on the street in Toronto of Vincent van Gogh in a hollow carving. But as you walk in front of it, when you face it head on, you might experience this illusion where suddenly it seems like the face is actually coming out of the statue and being convex rather than concave. And this is known as the hollow mask illusion, which has been shown in many different forms. And it's thought to be due to the fact that we are so experienced at seeing convex rather than concave faces. So we have this prior knowledge that faces should be convex, which overrides the fact that in this case we know it's actually a hollow carving. So prior knowledge that we have about the world influences the way we perceive it. So what we study, the way we study this in the lab is using much simpler stimuli. And what we and others have seen in multiple studies is that our prior expectations of what will happen next can bias our perception. So for instance, a cue that predicts the likely direction of motion of a cloud of dots biases people's perceptual reports, as well as the representations in the visual cortex of the motion direction of those stimuli. The question I want to focus on today is, can expectations make us see things that are not there at all? I'm going to start with a sub-question, which is, can expectations on their own evoke a template of the stimulus that you expect to see in the absence of any input? And in this case, specifically, we'll be looking at the different layers of the visual cortex. And that's because, as Matthew also already alluded to, we know that bottom-up input into the visual cortex arrives in the middle layer, layer 4, whereas feedback targets the agranular layers. So if we're studying an internal process versus an external input, distinguishing the different layers can tell us something about the origin of these signals. The way we do that is we scan activity in the visual part of the brain using 7 Tesla fMRI and 0.8 millimeter resolution voxels. So this allows us to go beyond just studying the cortex as a whole, which in the case of the visual cortex is about 2.5 millimeters thick, which is about the size of a typical voxel. And now we have about 3 voxels that divide up the cortical sheet. We then separate the 3 cortical layers, so superficial, middle, and deep, using a spatial regression to try to deal with the fact that, of course, no voxel is ever perfectly only in one of these 3 compartments. There's always a partial volume issue here, so we use a regression to account for that. Okay, the study was led by Fraser, and also who was a postdoc in the lab at the time, and also with help from Oliver, who is a PhD student in the lab currently. And what we did is we had cues that predicted the orientation of an upcoming grating with 75% validity. In this case, the cue was the color within the fixation bullseye. So cyan might predict a right-tilted grating and orange a left-tilted grating. The first grating was followed by a second one, and people had to do a discrimination task on the 2 gratings that was unrelated to the expectation cue. So that's just to get them to pay attention to the gratings, but it's not relevant to our experimental question or to the to the predictive cue. Now, the crucial trials here were these 25% omission trials. So in these trials, there's still a cue that predicts a specific oriented grating, but the screen stays empty except for the fixation bullseye. So now there's no signal coming in from the eyes into the visual cortex. So any signal that's there must be due to other internal connections. So it's similar type of logic as the studies that Matt described earlier. Okay, so we were specifically targeting V1 in this case. So we used an anatomical mask to define V1. And then within that anatomical mask, we selected voxels that responded to our gratings using an independent functional localizer with flickering gratings. Within those active V1 voxels, we then selected the 500 voxels that most strongly preferred left-tilted gratings over right-tilted ones, and vice versa. So now we have 2 sets of orientation preferring voxels. And for those voxels, we determine how their volume is distributed across the 3 different cortical layer compartments, as well as white matter and CSF. And we use our regression approach to separate the laminar signals. Now, what we do next is we subtract the signal in the voxels that don't prefer the current orientation from the voxels that do prefer it to get an orientation-specific Bolt signal. So what does that look like? Well, for the 75% of trials where a grating is actually presented, all the 3 different layers in V1 show this orientation-specific Bolt signal. So this is kind of analogous to decoding the orientation of the grating. So the signal is present in all 3 cortical layers, as you would expect when a stimulus is actually presented to the eyes. What happens on these omission trials where people have an expectation, but no stimulus is actually presented. On those trials, we only see an orientation-specific signal reflecting the expected grating in the deep layers of V1, not in the middle and the superficial layers. So this has a very, very different laminar profile, with only the deep layers representing the expected stimulus. And this is a strikingly similar pattern to one we found a few years previously using a very different paradigm, namely the Caniza illusion. You can see that the illusion figure is quite small here, but basically we looked at voxels that have a receptive field on the illusory triangle caused by aligned Pac-Man. And we're careful to only select voxels that overlap with the illusion and not with the Pac-Man themselves. And in that study, we also found an increased activity for the illusory figure only in the deep cortical layers of V1. So there seems to be some similarity in the laminar profile of these two very different feedback effects. Okay, so that's the answer to question one. Expectations can activate what we might call perceptual hypotheses in the deep layers. So an expectation of what might come next, or a triangle you infer to be there because of these aligned Pac-Man. Now back to the main question, can they make us see things that are not there at all? Because in this omission paradigm I just presented, people did not hallucinate gratings there. It was very clear when there was a grating on the screen, it was very high contrast and was on there for about a second, versus when the screen was empty. So Joost has been leading a study where we try to investigate this question specifically. In this case, the cues were auditory cues that predicted the orientation of the gratings. And then either a grating would be shown embedded in noise, or only noise would be presented with no grating in there. So the gratings were much harder to see because of the noise. They were also flashed much more briefly, just 33 milliseconds. And I should point out that these noise patches were selected. We selected 4 noise patches that had no orientation-specific information in them themselves to rule out any stimulus-driven percepts here. After the stimulus was presented, people were asked to report which orientation they thought the grating had and how confident they were that there was a grating on that trial. And again, as in the previous study, a separate functional localizer was used to select voxels. Okay, so let's have a look at what we might expect to see here. So what happens when we have these kinds of hallucinations? Keeping in mind the deep layer effect we found before. It might be that on trials where people hallucinate grating and noise, that is, again, specifically the deep layers that are involved. So these perceptual hypotheses might be what's really truly important for subjective perception. And on trials where people don't hallucinate the expected grating, maybe there's no signal at all. Or the deep layer effect might be there regardless because people expect this orientation. But to actually also perceive that grating, maybe the superficial layers need to be involved, which feed forward signals back up the cortical hierarchy. So maybe both those components are required. And these were our hypotheses going in, and as you'll see from results, these hypotheses were both completely wrong. So let's have a look at the behavior first. People were more accurate in high confidence trials than low confidence trials, as you might expect, and they were more confident that a grating was present when a grating actually was present than on the grating absent trials. But crucially, confidence on those grating absent trials wasn't at floor, and there was quite a spread. So here we see the 4 different confidence responses, 1 through 4, and you can see that also on the absent trials in orange, there was quite a spread, and there's still quite a few high confidence responses, even when there was no grating present. So here we just split the low and high confidence trials, and we see that in about 35% of trials, people have high confidence that a grating was present, even when there wasn't one. We also debriefed them afterwards, so we didn't tell people beforehand that there would be noise-only trials, and we trained them in a way where we started with 100% grating present trials and then slowly made the gratings noisier and noisier and started introducing these noise-only trials. And we debriefed them afterwards. 50% of the trials actually were noise-only trials, and only one person realized this. All the others were at far lower estimates of how many trials they thought contained only noise. So what about the effect of the auditory cues? Did people hallucinate or falsely perceive the gratings that were predicted by the auditory cues? Well, if we split our participants into, based on debriefing, on those that became aware of the meaning of the auditory cues and those that did not. So in this study, we did not tell them what the auditory cues meant to avoid response biases. Then if we first focus on the people who did not realize what the auditory cues meant, there's no effect at all of the auditory cue, the prediction induced by the auditory cue. So they were just as likely to report seeing the grating that was not predicted by the auditory cue as the one that was predicted by the auditory cue. Only for a few people who did become aware of the auditory, what the cues meant, 3 out of the 7 people who became aware of it, was there a strong effect of the cue, and we put this down to being likely due to more of a response bias or strategic guessing rather than an actual perceptual effect. But regardless, also if we group over the whole study, over the whole set of participants, there's no real effect of these predictive cues. So what we conclude from behavior is that people have these false percepts. They have quite a lot of high confidence reports that they saw a grating even when there wasn't one. But which grating they saw is hardly, if at all, determined by the predictive cues that we presented. So that was surprising to us, of course. What about the neural effects? First of all, as in the previous study, we found that only significantly in the deep layers did we find an orientation-specific bold signal for the expected gratings. So in this study as well, the deep layers represented the expected but omitted orientation. In this case, it's in V1. These gratings were lower spatial frequency. That's probably why. I'm happy to discuss further if you have any questions. Now, what about these false percepts? So first, the low confidence trial. So here they were forced to report an orientation, but here they said there probably wasn't a grating. In that case, there's no orientation-specific bold signal reflecting their guess in any of the layers of V2. But on the high confidence trials, there was, specifically in the middle layers of V2. So in the middle layers of V2 here, there was an orientation-specific bold response that reflected the grating that people hallucinated in noise, if you will. So this was a surprising effect. As I said, it's not in line with either of our hypotheses. We assumed the feedback layers to be involved in this, but it actually seems to be the middle input layers that are crucial for these false percepts. So prior expectations can bias our perception and evoke stimulus templates in the deep layers of the visual cortex, but to actually see things that are not there, it seems that the input layers, the middle layers, need to be involved. So this raises a lot more questions for us than it answers. So one of our questions is this what happens in actual hallucinations? When people have an actual hallucination, is there somehow the activity in the input layers involved? Is it overwritten somehow? Well, we don't know the answer to that, of course. One data point that we think is encouraging is that we did a separate online study and found that these the confidence people had in these false percepts on average correlated with how likely they were to have abnormal percepts in daily life as determined by a questionnaire. So there is at least some correlation between these kind of arbitrary, abstract, lab-based percepts that we induce and abnormal percepts people have in daily life. So the question we have now is, or one of the questions is that maybe certain priors or priors in certain populations can unlock the input layers and even in non-clinical populations, for instance, for indirect connections from the deep layers to the middle layers. Okay, so I wanted to change tack for the last part of my talk if there's time. I don't know how I'm doing for time. Okay, great. Yeah, I'll just go to the last part very quickly. So this was the main thing I wanted to talk about, and as I said, this raises a lot more questions for us than it answers, but I hope you'll agree that it seems to be very relevant and interesting to look into these layers, even at the relatively coarse level that we can do so in humans, because without this layer specificity, we would have drawn the conclusion that signals induced by expectations and these false perceptions were the same if we just average over the layers, but actually by looking at these cortical layers we see that they actually are quite different signals and likely result from different sources. So in a separate line of work we look at where do these expectation signals come from, and our main contender, the main region we look into is the hippocampus. Our hypothesis is that when you present a predictive cue, the hippocampus, through pattern completion, might retrieve the associated prediction and send it to, in this case, the visual cortex. And in some previous work, we found that we can decode from the hippocampus what the predicted, in this case, our abstract shape is on a given trial. And in recent work, led by Fraser again, we've tried to look into how these predictions build up in the hippocampus. And what Fraser found is that, kind of surprisingly, as people learn, so this is while they learn while in the scanner, they're not pre-trained, then towards the end of these blocks of 32 trials, we start to be able to decode from the hippocampus only unexpected shapes. So expected shapes in green are not significantly represented in the hippocampus, only unexpected shapes. So this looks kind of like a prediction error signal. Which is so a kind of a negative prediction signal, which is very different from what we found previously, as I showed in that whole brain searchlight result, represented by the square here at the end. So how come we found a prediction signal in the hippocampus when we pre-train people, but a prediction error signal when we train them in the scanner? Well, our hypothesis was that maybe the hippocampus emphasizes different signals depending on the stage of learning and how informative predictions versus prediction errors are. And if this is the case, then we would expect hippocampal representations to switch sign over learning if we expose people to these regularities long enough. So what we did is we just made the blocks longer, 128 trials per block here, and we found that indeed, specifically in the posterior hippocampus, there's a switch in the sign of these prediction signals in the hippocampus. Early on in learning, only unexpectedly presented stimuli are represented in the hippocampus, and validly predicted stimuli are not. But later on in learning, it's the predicted stimulus, regardless of which one was actually presented, that is decodable from the hippocampus. So I'm going to skip this last data point. So what we conclude from this is that the hippocampus switches from representing errors to predictions as learning progresses and novelty and uncertainty reduces. Future directions include looking at the direction of communication between the hippocampus and the visual cortex, looking, for instance, at the layers of the entorhinal cortex. All right, I would like to thank the people involved in this, especially Joost, Fraser, Oliver, and Georgia, who did all the work, and MR physics team at the Welcome Center, led by Martina Callaghan, without whom none of this work would have been possible. And thank you for your attention.
Yes, thank you very much. There are quick time for a few questions. So, was confidence related to the hallucination effect on the trial-by-trial level?
I'm not sure what you mean by trial-by-trial level. We just split the trials based on if the confidence on that trial was higher than average for that participant or lower.
In what cases do you see feedback effects in the superficial layers, like Lars, and in what cases you see feedback effects in deep layers?
Yeah, great question. So I think one of the things I'm wondering about is what the role of attention may be. So these predictions that we looked at here were implicit. People didn't know what the cues meant. When we look at working memory studies, for instance, where people keep a grading in mind vividly and in order to do a task, then it's both deep and superficial layers that are involved. So I'm wondering if there's an attentional component there. When attention is involved, the superficial layers also become engaged. But that's very speculative.
Okay, Peter, did you consider aphantasia or measure ability for mental imagery?
I did not, but I think that would be a great idea. Yeah, I didn't consider it here, no.
And finally, does the layer segmentation assume equi-volume layers? And if so, how well does the mid-layer correspond to layer 4?
Yeah, great questions. Yeah. So by the way, coming back to aphantasia, we're working on some questions with Nadine Dijks that to do with imagery and also considering layer experiments. So we may get to that in the future. But yes, they're equivolume layers, so they're equally thick layers in the 3 compartments. And for V1, based on histology, we know that this roughly corresponds to layers 1, 2, 3, 4, and 5, 6. For other regions, it's maybe less clear-cut, but for V1, it seems to be relatively good, work out relatively well for us.
Yes, thanks a lot. They're very good questions. I'm really enjoying that, although the rating makes it jump up and down. So thanks for the question. Let them keep coming. Thanks for the answers, Peter.
Thank you.
And now we move on to our final speaker, Maria Wimber, with the title, A Flip in the Representational Hierarchy Between Perception and Episodic Memory.
Okay, great. Yeah, thanks a lot, Lars, for putting this together. Really great symposium. Very much enjoyed the talks before. My name is Maria. I'm the local in this local organizing symposium. My lab is based at the University of Glasgow here, and we study memory. So the question that really pretty much everyone in my group is interested in is how does the human brain reconstruct episodic memories? So memories of single-shot learned, unique events in your own autobiographical past. If I ask you, for example, what you did last weekend, I guess I was preparing an OHBM talk, but there might be a more enjoyable thing to report, like you went on a nice walk with your family, then went to the pub, enjoyed a nice meal, and so on. So this would be a typical example of an episodic memory, and we're interested in how the brain does that. How does it bring back to mind these very vivid, multi-layered images, or not just images, integrated memories of past events? So in our field, we have very solid, very good computational models that all sort of go back to David Marr's seminal work in the 70s, but also hippocampal indexing theories or complementary learning system theories that kind of make the common assumption that when you experience an event, your neocortex will record the ongoing activity that is, or it will represent the ongoing activity associated with that event, and the hippocampus will continuously store what is called an index to what's going on in the rest of your brain in neocortex, right? So it binds all of this information together such that at a later point in time, when you get a partial reminder that overlaps with the original event stored in memory, this will ping the hippocampal index, and the hippocampal index or the hippocampus can then go and back project into neocortex. It kind of unzips the information that it previously associated with this reminder. So the minimum prediction from these models is that the patterns that we see during encoding, seeing here on the left, should overlap to a big degree with the patterns that we see during retrieval. So neocortical activity should look very similar here. So what we are very interested in my lab is this really true? Are memories really just snapshots of the events that we previously experienced? Or is there some sort of prioritization of what information is contained in a memory? We're also very interested in how this process of reconstructing a memory unfolds in time, where these models don't actually have a lot to say. They kind of see this as an all or none process, but we're very interested in whether this is a layered process that actually evolves over time, and also how remembering in itself then changes the underlying memory landscape, although this is not something I'm going to go into today. Right, so let's move to these two questions. What features are preferentially coded or contained in a memory, in a reconstructed memory, and how does that process unfold in time? We took inspiration from all these decades of beautiful work that's been around from the vision literature, some of which we've heard here, where we know that, at least for relatively simple object images, once these images enter the brain from the retina and hit neocortex, low-level visual areas will start analyzing low-level visual details of the information, and then the information travels forward along the ventral visual stream, and along this pathway, it becomes more and more abstracted, so that regions that really sit close to the hippocampus code relatively abstract, sort of just like features. They code actual concepts, like these famous Jennifer Aniston neurons, for example. So we think the hippocampus receives this highly integrated and very highly preprocessed abstract semantic signal. So for all of the experiment I'm showing you here in the first talk, the very simple working hypothesis was, well, if the hippocampus then goes back project into neocortex, it should do so in a reverse order from what we see during original perception, right? So we should see that first the hippocampus, or via the hippocampus, we recover semantic abstract information, and then the brain kind of works backwards to more low-level visual information. And so the kind of paradigm that goes throughout the rest of this presentation is really just a very simple associative learning paradigm where we always use a reminder that is linked to a certain image. At a later point in time, I give you the reminder word, and I ask you to reconstruct the image linked to that reminder in as much detail as you can, and then to press a button as soon as you have a vivid image back in mind. The twist that we're doing here is that we manipulate these images along certain predefined sort of orthogonal dimensions. And we always have one perceptual dimension that is on a lower level visual level. So in this case, for example, an image could be presented either as a photo or as a line drawing. And the idea is, if a region really cares about this lower level perceptual format, it would group together all of the photos and all of the line drawings, irrespective of the actual content of these images, the meaning of these images. And then we always have a higher level semantic category where an image can, in this case, be, for example, an object or an animal, like animate or inanimate. And again, the idea is if a brain region really cares about semantics, it should group together all of the animals and all of the objects and not care so much what the perceptual format is in which these images are presented. Right, so the first data I want to show you is from an EEG study where we were interested in how this information unfolds in time when people are either seeing information on the screen. Let me just see, is that a laser pointer that I can use? Here or on the screen? Yeah. Yeah. So we were interested in how this process unfolds in time when the image is either here on the screen or there's nothing on the screen and people are just mentally reconstructing these images from memory. And what we did here is train two different LDA-based classifiers, one perceptual that was supposed to discriminate the perceptual format, and one conceptual classifier that we asked to discriminate between the two semantic classes in our imaging set. And then we trained these classifiers on each time point in the EEG data, the sensor patterns serving as the features, and we kind of let the two classifiers race against each other, right? So we asked, does the perceptual classifier peak before the semantic one or vice versa? And the idea is, if there really is a forward stream of information processing during vision, we should see the perceptual classifier peaking before the semantic one here. If this process is flipped during memory recall, we should see the reverse pattern down here, with the semantic being followed by the perceptual classifier. So when you look at the average decoding peaks, which is just when does a classifier peak on average across the trials in a single subject, we already start seeing this pattern. It's not quite significant for the forward pattern vision, but quite a strong significant effect that semantic information during memory recall comes back around 200 milliseconds earlier than the lower level visual information. You can also see this effect if you go down to really the single trial level. So you take a single trial classification timeline and see where is the peak of the perceptual and the semantic classifier. Things become a bit noisy, but there is a strong significant shift towards blue, which means perceptual before semantic for perception, and there's a strong shift towards red, which means semantic coming before perceptual information during memory retrieval. You can even see this in simple ERPs if you just compare animate and inanimate, or photo versus line drawing, so you don't actually need to do classification on the data at all. It's quite a robust effect. And so we wondered next, well, if we see this in relatively noisy EEG patterns, can we turn our human participants into classifiers? Can we see this effect in the reaction times and behavior? And so we modified the task a little bit. We brought in a visual group who would, on each trial, really simply see one of these objects on the screen, but before the object comes on, they are asked either a semantic or a perceptual question. So is the upcoming object a living or a non-living thing, or a photo or drawing? Object comes on, you press the button as fast as you can. And then we do the same thing with a memory group who have previously been trained to associate these reminders with the objects. So they never actually see the objects in this phase, reconstruct them from memory, and the question is the object that was associated with that reminder a living or a non-living, or a photo or line drawing? And people again press the button as fast as they can. And so what we've replicated now, I think we've got more than 10 experiments by now, is that this is very strongly visible in just in behavior and reaction times. So you see, shown a little bit differently here, that whenever the information is on the screen, people are significantly faster at classifying the perceptual than the semantic features. And then this flips around when people are reconstructing this exact same information from memory. We classify the semantic features faster than the perceptual one. The only experiment so far we have where this doesn't work is when the perceptual feature is confounded with semantics. So here we used shape, and I think because a turtle is always naturally round and a spoon is always stick shaped, you get a confound between semantics and perceptual features here and don't find that effect with it, which is quite interesting in itself. Okay, so then we were wondering about the locus of these effects, because our hypothesis is there's a backflow of information along the same processing pathways that also carry forward the information originally during perception, and we just wanted to see whether this is actually true. So we did, this was actually a simultaneous EEG fMRI study I'll show you in a second, but let's first look just at the fMRI data. So again, what you're seeing here is the two different decoders, and not surprisingly, we're able to decode the lower level perceptual information from earlier visual areas than the higher level semantic information when these images are on the screen. For retrieval, I think the most striking effect that we see really is that everything seems dominated by semantics. So you can decode semantic information much more easily when people are generating this information internally. There are some clusters of for low-level visual information as well. You also see that this involves some of the areas that were originally part of the encoding pathways, but also additional areas here in anterior temporal lobe and in frontal lobes. And so we next were wondering, as I maybe say, so we had in-scanner EEG data there. Originally, the plan was to fuse these two data together from the same subjects, but the EEG in the scanner really didn't look that great. So we went and took the out-of-scanner EEG data set that was recorded in different subjects, and we attempted a fusion of the EEG and fMRI data, very much in the same way that Matt actually explained before, so similarity-based fusion, like people are doing between monkey and human data, for example. So we take the EEG signal. For each time point here, you can get the pattern, the sensor pattern in response, for example, to the octopus, to the camera, and then you can start filling a similarity matrix with these pairwise similarities. You can do the same thing for your fMRI signal. Again, fill your matrix for the fMRI, and then now we beautifully have this information for the EEG and fMRI in the same format, and we can simply do, whatever is your preferred similarity metric, Euclidean distance, Spearman correlation rank, and so on. And so when we do this for encoding, maybe again, not surprisingly, you see that the information, so we do this time point by time point here, right? And then you see that similarity between the EEG and the fMRI signal starts emerging in early visual cortex, surprisingly late. I don't have an answer for why, or not a good answer for why it is so late. But then you see the information progressing forwards along the ventral visual stream until it peaks around 700 milliseconds, and then it recedes again after this time point. No big surprises here. For us, it was more interesting to see what's happening in the retrieval data, building up again to this time point when people press the button to indicate that they've got the memory back in mind. What you see here is that the similarity structures start matching about 2 seconds before the button press, really neocortical areas around the hippocampus. Then we don't find a significant cluster for about 500 milliseconds. And then we do see these sensory areas coming back, but really the large bulk of the information that we're decoding here, or that we're getting here, comes from more parietal structures. So this is the kind of backward stream that kind of goes back here, but also really projects into these medial and superior parietal areas. So to conclude that part, and then I have a shorter one coming, we do have evidence that the information flows forward and partly backwards along the same trajectories during memory recall, but this is clearly not the whole story, and we're working a lot on trying to figure out what the transformations are that explain these, this progression to parietal areas during memory recall, and what kind of information is actually carried there. So I guess the other question you can ask then if the information at least partly flows along the same neural pathways, how does the brain keep that separate? And I guess one answer has just been given by Peter, who showed nicely that the information flows in different layers. So we can assume that it also flows, or we know actually from really beautiful work by David Barron, for example, it flows into the hippocampus and out of the hippocampus in different layers. But there's also extremely elegant models, computational models that go back to John Lissman and Mike Hasselmo that assume that the hippocampus has this temporal code where it keeps information separate in time, actually. So the idea here is that a theta oscillation in the hippocampus clocks the information processing pathways such that at the peak of this theta oscillation, the gateways from the entorhinal cortex into the hippocampus are very open, so this is good for being in an encoding state. Whereas at the trough of this theta oscillation, these gates are relatively shut, and the hippocampus engages more in this auto-association, pattern completion process, and puts information back out into neocortex. And we were wondering, there's beautiful rodent work already showing this, and we were wondering if we can also observe this in humans. And so for this study here, we used EEG again and the conceptual classifier, simply because that works better than the perceptual one. And we looked in more detail at these single trial classifier confidence traces. So we see this as an index of the reactivated memory content, right? And we were wondering first, well, is there any rhythmicity in these traces? Is there an oscillation, basically? The kind of, or do they go along in an up and down, waxing and waning sort of fashion? You can do this as a first simple step by just taking these classifier traces and feeding them into a Fourier transform. And when we do that, we see there is a strong significant peak around 8 Hertz, which is a relatively fast theta frequency. Importantly, this exceeds a noise threshold that we're fitting here from feeding the exact same data, the raw data, to the classifier, but not giving it meaningful labels. So we think this up and down really has to do with a memory reactivation process, recovering meaningful information, and not with the raw signals that we're feeding the classifier. And then the second thing you can ask here is, well, if we identify the peaks of this classifier during memory, but also during vision, do these peaks fall onto different phases in a theta oscillation, which we source localize as best as we can with EEG data here to the hippocampus? So when you look at that, you see a really strong phase shift such that both the encoding and the retrieval traces are modulated by a theta oscillation, but the information that's recovered during memory recall sits on the opposite, almost 180 degrees phase shifted portion of the cycle than the information during actual visual exposure to these stimuli. So there really seems to be this temporal segregation of information consistent with the Hasselmo model. Right, let's see, can I do 3 more minutes or so? Are we fine? Yep. Okay, so last empirical evidence there. Marai Terval was then wondering, inspired by a lot of studies coming out of the attention field at the moment, can we actually see this rhythmic modulation also in behavior? And we had so many behavioral data acquired at this point that we had loads of button presses on these memory tasks and the vision tasks from 266 participants at the time. And she then went and used a method that is called the oscillation score, which is really a method for detecting oscillations in spiking patterns, usually. She applied these to the response traces from our human subjects doing these memory and vision tasks. So obviously, so this is an example of a trace from one single subject. You see, very typical for response time distributions, that you have this left skewed, long right tail pattern, but we get rid of this overall distribution and we're just interested in these little ups and downs that are contained in the slower overall form of the response traces. And if you formalize that using the oscillation score and also simulations that make sure that we don't detect too many false positives here, the most interesting observation is that we find significant evidence for an oscillation in behavior during all the phases that depend on memory, but not for the phases of our task where people just do a simple visual classification, or they've already completed the memory task and just have to answer one of these catch questions. And then just as a proof of principle, so we're also seeing these oscillations most dominantly in the lower theta range. Marai then also used intracranial data, recordings directly from the human hippocampus in epilepsy patients. And she showed that exactly in these task phases where we also find the behavioral oscillations, we also do find a very strong and temporally extended theta phase locking that can explain this rhythmicity in behavior. Right, so I guess the very short take-home message is we do think memories are, at least on the temporal level, reconstructed in reverse order with a prioritization on semantics, and they're clocked by the hippocampal theta rhythm. I'd like to thank all of the members and former members in my lab, and especially these 5 people here, who are the masterminds behind all of the studies that I've shown you here. And if you give me one more slide. Our local imaging center here, the CCNI, the Center for Cognitive Neuroimaging, will be organizing a party tonight where everyone is very heartily invited. I know it clashes with the postdoc and student social, but I think this will be a lot of fun. We have an open stage, open turntables if anyone is inclined to go up and perform. Entry is free. We have a very generous open bar as well, and you just need to pull a ticket by taking a picture of this code here and join us tonight from 8 p.m. in the West End in the Oran Moor. Thanks a lot for your attention.
Well, thanks a lot. We have time for a few quick questions. Maria, do you think lower to higher order might also indicate an external visual versus self-relevant default mode network gradient of organization?
Yeah, I'm pretty sure this is what we would see if we map the representation, or what we do actually see. So if we just ignore time and map the representational structures onto the different brain networks, I think we would perfectly see more default mode network information carrying the mnemonic, internally generated content, and the more sensory, externally driven pathways carrying the visual information. So I think, yes.
Concerning the perception dichotomy, can we consider drawing versus photo also as a semantically meaningful category, real versus artificial?
I'm not sure, to be honest. So, I mean, it's obviously a meaningful distinction, and I can put a word, like I can verbalize if something is a photo or line drawing. But then I find it very hard to find a very low-level visual category that is not verbalizable. So I guess even in V1, where neurons really strongly respond to orientation, you can put a word on that. You can say left or right. So it would be a meaningful distinction, but it would be lower level than the semantic labels we use for animate, inanimate, for example, or exemplar level categories.
The semantic areas identified in the fMRI data are more posterior and distant from the hippocampus than expected. Why?
Yeah, I do not know, but there's anterior temporal lobe and areas around Broca, obviously the most studied areas in the field of semantics, and we're doing a lot of work at the moment seeing how prior semantic knowledge about things influences memory recall. So I think these probably carry those kinds of semantic representations that have been engraved into your neocortex in a statistical learning kind of way across your whole lifespan, and that's what we're seeing here. It's probably not the one-shot patterns that we imprint by just the images they see in our tasks.
And during retrieval, could it be motor preparatory activity?
Say again?
During retrieval, could it be motor preparatory activity?
I mean, there will definitely be a component, because whenever decoding EEG or MEG data, whenever you have an ERP component, you're better at decoding information. So I think the boost in decodability building up to the button press itself maybe is at least temporally aligned or facilitated by the motor response, but I don't think motor activity can explain the differential decoding of the semantic and perceptual information.
All right, perfect. Now I'm crashed the app anyway. We are out of time. Thanks everyone. Thanks the speaker, and thanks you for coming to this symposium. And now we have the lunch break.